Hello, everyone. This is Yoshinori Hayakawa reporting from Creation of Eternal Life spread over unlimited and eternal universe by science and technology. My document can be seen from this PDF file address. The creation of eternal life spread over unlimited and eternal universe by science and technology. Researches on biological point of view. This was presented at Dalian, China, in April 2014. The first, I will report about the reasons of extinction of life. First, nuclear war and nuclear winter. Second, global climate change. 3. Deterioration of human genes. 4. Large meteorite or comet impact. 5. Intensification of eruption. 6. Increase of sunlight. 7. Gamma ray burst in near star. 8. Change of the sun to red giant. 9. Approach of Neutron Star. And 10. Change of the sun to White Dwarf. First, nuclear war. Nuclear war and nuclear winter. First, I will report about nuclear war and nuclear winter. There is a famous principle for peace. 5 principles of peaceful coexistence. It was agreed with China, with India. 5 principles of peaceful coexistence. Mutual respect for sovereignty and territorial integrity. Mutual non-aggression, non-interference in each other's internal affairs. Equality and mutual benefit, peaceful coexistence. It was agreed between China and India in 1954. It was reported in China. It is written in Chinese. Yes. If this 5 principles of coexistence is maintained, there will be no war and no nuclear weapon, no nuclear war. But presently, the world situation is changing, and we cannot guarantee that there is no nuclear warfare. Second, global climate change, warming or cooling. I report first in this red line. Second, yes. When the sunlight is increased, we are going to reflect part of solar energy light by a satellite mirror. This is the sun, this is the earth. Satellite mirror or increasing carbon dioxide. Third, deterioration of human gene. Keep present human DNA frozen and protected from radiation and recover to human when human gene is denatured. Due to medical development, those who are to be perished 100 years ago in use can survive and leave descendants now. It is a victory of science, but at the same time, not appropriate genes in the past are left for future generations. If our civilization collapses by some reasons, for example, lack of fuels, then we cannot keep our civilization. Our civilization collapses and the world became agriculture based and manufacture factory. All human beings will be perished. Deterioration of human gene. 3.1. Deterioration of nuclear DNA. Keep present human cells frozen and protected from radiation and recovered to man by the exchange of nuclear DNA with that of living human male cells when human gene is deteriorated too much. We have to establish this technique somehow. We are going to use this technique later. Probably we seem to use similar technique of making iPS cells, and in children born by marriage with future person, number of deteriorated genes become half. Recover of live mammals from frozen cells and live elephant cell is planned. Deterioration of human gene. 3.2. Deterioration of mitochondrial DNA. Mitochondrial DNA is transferred from mother and not from father. So take gorilla, chimpanzee, and or orangutan cells that are not infected by dangerous virus and exchange their nuclear DNA with that of humans and investigate if it is possible to recreate humans. Life thus created is considered as humans. Key primates such as gorilla, chimpanzee, and or orangutan eternally without medical care, but keep them not infected by dangerous viruses. Their mitochondrial DNA will not be deteriorated like that of humans. 3. Total deterioration of DNA. Frozen nuclear DNA is transferred to live human cell, and half of the male and female population is created. Nuclear DNA of living human cells are changed with that of living cells of gorilla, etc., and another half of the male and female population is created. Thus, the deteriorated DNA becomes half for both nuclear and mitochondrial DNA. 3. Deterioration of human gene in long term. Assuming point mutation and deterioration of DNA of ancestors and gorillas are negligible. D. Deteriorated DNA during certain period. T. AN. Deteriorated DNA after NT period without repairing process. AN. ND. A. Infinity. Infinite. BN. Deteriorated gene after nth repetition of the repairing process using old ancestors' nuclear DNA and gorilla's mitochondrial DNA with time interval of T. Humans with repaired DNA include half D and another human without repaired DNA have D. 3. Deterioration of human gene. The son of or daughter of them have B1 3/4 multiplied by D. Similarly, after second repair, B2 equals 3/8 plus 3/4 D. B3 3/16 plus 3/8 plus 3/4 multiplied by D. This is the geometric sequence of the ratio of half, and hence B infinite will be 3 divided by 2 multiplied by D. Just previous repair is previous to repair is D higher. The future degeneration is between 3 second multiplied D and 5 second multiplied D. 4. Large meteorite or comet impact. That happened 65 million years ago and perished the dinosaurs. That can be avoided by evacuation to space colony, moon colony, Mars colony, and Space Growth Center is going to change the trajectory of large meteorite or comet from collision to Earth, but it is not yet successful. Intensification or eruption that happens 250 million years ago. Also, we can evacuate to space colony, moon colony, and Mars colony. And 6, yes, this is perishment of dinosaurs by large meteorite collision. This is where we are going to evacuate. Yes, space colony. Six, the reason of extinction of life, increase of sunlight. Within 100 million to 1 billion years, life cannot live on the surface of on Earth because the sunlight becomes stronger. And to evacuate that one, we are going to reflect sunlight by satellite mirrors and evacuate to space colony and underground colony, Mars colony. This is a famous Hertzsprung-Russell diagram and future of the Sun. This is a surface temperature, and this is the magnitude of the star. This is the main sequence, and the Sun is on this line. At present, Sun is here, but solar energy is increased. And then nuclear fusion in solar Sun are going to change, and it will become a red giant, and then 5 billion years later, it changed, finished nuclear fusion, and 10 billion years later, it will become a white dwarf. And no life can live in the solar system. Increase of sunlight. Sunlight increases gradually, and no life can exist on the surface of Earth 100 million to 1 billion years later. To avoid that, satellite mirror to reflect sunlight, evacuate space colony, Mars colony, underground colony. Reflect part of sunlight by mirror satellite. This is the Sun and this is the Earth, and part of the solar energy is reflected by satellite mirror. This is the evacuation to space colony. This is a picture of space colony. Living long time in space colony is required. Only utilizing solar light energy and establish nearly zero discharge life system. Euglena create food and oxygen gas from water, sunlight, and carbon dioxide gas. Japanese company has succeeded mass production Euglena and seems to be useful for establishing nearly zero discharge life system in space colony. This is a picture of Euglena. It's a unicellular organism, and it is an animal and at the same time plant. This is the eye of Euglena, and it has a color of green to it's showing that it is a plant, so it can produce nutrition from sunlight, water, and carbon dioxide. Large-scale production is established by Japanese company. The site can be seen from this site. Reason of extinction of life. 7. Gamma ray burst in near star, that it can be avoided by evacuation to underground or under sea level base. Change of the sun to red giant happened 5 billion years ago. Evacuate to a space colony far from the sun, Saturn satellite colony, voyage to other star system. This is a previously shown health spring Russell diagram for and future of sun. 5 billion years ago later, sun will become a red giant, and sun's nuclear fusion is changed and it becomes very large, and this is Earth and Jupiter and Saturn, so no life can live on Earth. Evacuate to space colony. Evacuation to space colony far from sun, or evacuation to Saturn's satellite base. 8.2. Using only solar light energy and establishment of nearly zero discharge life system. Probably Euglena is useful. B. Transferred to other star by computer-controlled spaceship in the form of DNA and later recovered to life by automated manner. 9. Reason of extinction of life. Approach of Neutron Star. Neutron Star is much heavier than the solar system, so solar system is destroyed because of big gravity of neutron star. Yes. This shows how the Earth is destroyed by neutron star, but where it will approach to solar system is very difficult to find because neutron star does not emit light. change of the Sun to white dwarf 10 billion years later, all nuclear fusion is stopped and the Sun will be allowed to cool down. Approach of Neutron Star transferred to other star by computer-controlled spaceship in the form of DNA and later recovered to life by automated manner. Red dwarf has less mass but longer life than the Sun, so it is better to evacuate. Nearest star to the Sun is 4 light years apart. If the spaceship moves 1 part per 10,000 of light velocity, it takes 40,000 years, while the universe is 270 degrees minus 270 degrees in Celsius, and no light energy from star is available. So it is impossible to keep many life alive during voyage as sufficient energy source does not exist. B. Therefore, life are kept as frozen cells during this voyage in computer-controlled spaceship. Josephson computer or quantum computer seems to be suitable as they function at very low temperature. As the spaceship approaches the targeted star, frozen cells are recovered to life automatically. Computers and software should not make mistake at least 40,000 years, if possible, several 100,000 years. During that period, that will not mistake B. Keep DNA several 100,000 years without denaturing. Frozen cells in nearly minus 270 degrees in Celsius and shield it against galaxy cosmic rays. Find method to automatically recover life from frozen DNA. C. Find method to automatically recover life from frozen cells. C1. Living cells seem to be necessary to recover life from long-time frozen cells. Small amount of living cells are transferred in spaceship. C3. Frozen nuclear DNA is changed with that of living cells and then recovered to life. C. Find method to automatically recover life from frozen cells. For virus, freeze-drying is used for short-term conservation, but not suitable for long-term. Virus will be recovered together with living cells and again freeze-dried. Contamination by virus must be avoided. D. Living life transferred in spaceship. D1. Suitable life as animal is tardigrade, water bear, and as plant is moss. they can survive 8 years in dry and freezing conditions. For each 8 years, giving them water and suitable temperature and light will keep them alive, and tardigrade lives eating moss. This is a tardigrade. This is a moss. Survived 400 years below glacier, Canada National Geographic. D. Living life transferred to spaceship. The problem is if mitochondria and chloroplast of tardigrade or moss functions well with nuclear DNA of another species. Mitochondria necessitates approximately 300 proteins produced in nuclear DNA, and chloroplast of plants depends on species. Mitochondria know not the mystery mitochondria by Kuano Kodansha in Japanese. D. Living life transferred in spaceship. If tardigrade or moss are not suitable for all animals, probably not for all animals or for all plants, cultured cells of many species must be transferred. They are frozen and each 5 years brought to normal temperature and cultured again. By freezing and culturing, cancellation of the cells are avoided, and materials and energy for culturing is reduced. If they are frozen too long, they will die. Necessary investigation 1. For human healthy life, many life such as plants, animals, intestinal bacteria, and flora of the skin are necessary, and we have to find out at least one set of life that can live together. We have to find out how they can be recreated to life from frozen cells. Necessary investigation 2. Many animals necessitate many plants and bacteria. These should be researched as well. Roots of many plants are cooperating with fungi to obtain nutrition. This should be researched, and necessary fungi are transferred together with plants in spaceship. Necessary investigation 3. For mammals, artificial uterus seems to be necessary together with robot mother. For humans, teaching machine is necessary as well. We should determine order and the timing of recovering life. For example, plants are recovered first and animals are recovered next, because animals cannot live without plants. So many biological researches are necessitated. It can be started right now. Increase efficiency of education. For creating eternal life, much development of science and technologies are required. For that purpose, much essential knowledge is required, and raising efficiency of education is necessary. Basic education, reading, writing, arithmetic, and foreign language learning should be as efficient as possible. Language learning possible. The author has developed computer numerals. Yes, this can be found in this site. And Universal Literacy Alphabet. This site is no more available. For the purpose, Esperanto seems to be suitable as easy language. Discussion and conclusions. Many points for creating eternal life was discussed. Nearly zero discharge life system, which uses only solar energy, is stressed in long life in space colonies. Uranus seems to play an important role in the above. Transfer to another star is necessary for eternal life. Where small amount of living cells together with frozen cells should be sent by automatically controlled spaceship shielded against cosmic rays. Living cells are frozen nearly all time except periodical given light, mild temperature, and nutrition for short time to avoid cancellation. Save energy and material. To save energy and material. Nuclear DNA of frozen cells are changed with that of living cells to recover to life. Much more research should be made concerning the mutual dependence of life to make a closed set of coexisting life. Importance of education efficiency is stressed for development of science and technology development. Thank you for listening.